I'm the last one, so I also wanted to, I hadn't thought about it, but I kind of start with a disclaimer, because also the title, I think it points more towards where I plan to go rather than what I will really be able to cover in the presentation. It's about active matter, so I guess being the last speaker, I don't need to motivate a lot why active systems are important, how we get inspired by nature at all scales from birds down to motors. But then we also know how to produce active systems in the lab. And I mean, I will be mostly talking about colloids. So these are a particular experimental setup of Quinker colloids in which by an applied oscillating electric field, then you can generate an instability, and then they can move together in these very well organized structures. But also there are quite a number of interesting issues related to how we can control and produce well-defined collective outcome and tasks out of local rules. These are small robots that talk to each other, but we had the first talk today was also about robots talking to each other. This is also, I think, a very quickly growing area where many of the ideas of what I will be talking also will be relevant. As I said, at the colloidal scale, then we know in equilibrium how particles interact. When they are out of equilibrium, the type of interactions, the effective interactions, are qualitatively different. And as a result, that opens the possibility to generate, manipulate them into structures that are not possible in equilibrium. And I think that's really a new opportunity in this type of systems. And in a way, I will be touching on this part also a little bit with the hope of understanding more clearly how they interact dynamically, and not only which phases they can organize, but how they can move in response to this situation, and how, in which sense the collective response is different from the individual one. And I mean, this is just to motivate that these are a nice examples. If you put a particle in an active, in this case it's a liquid crystal, you can generate rotation, or you can also bring colloids together, create ensembles that then you can, these were done with, these are magnetic colloids with which under also an oscillating magnetic field, they can organize and then they can be moved and manipulated in a microfluidic device. All of this, if you want, within the potential that it has beyond the fundamental understanding, I think it's really what it came in the title of the talk. We want to talk about robots, about small robots, and the idea is then if we miniaturize how we can really use them. And actually, there is a part of it that, I don't know, it's more science fiction. I think there is this classic by now, this Fantastic Voyage, where they pretended that it was possible to miniaturize people or have miniaturized ships going through the blood vessels, into the blood. So there is really a question of how we miniaturize. It's still going on. I mean, I was looking around, and you find when they talk about how we can use nanoparticles to cure cancer, in some places they come up with this type of images, and you may wonder whether this is real DNA with an object like this in the nanoscale, whether that's possible, or what is this about. So in reality, though, there are quite a number of experimental realizations of how to create at this scale objects that can propel or can be manipulated. For example, these are particles manipulated with ultrasounds, or these ones are particles in which these are colloids that basically decompose oxygen peroxide and generate bubbles. The oxygen peroxide decomposes in oxygen. If the oxygen, because if it's sufficiently at high production rate, it is more like a jet, and that's really then a microjet. And even at smaller scales, I mean, we have been seeing how liposomes are used to drug delivery, so now we are moving towards the nanoscale, and by also combining them with enzymes, it's possible also to make them propel. So I think that's an interesting potential, the possibility to really control the motion and the deposition of matter at small scales. As one goes from the micron towards the nanoscale, there are serious fundamental problems. Noise becomes very relevant, and as a result, a single of these objects can essentially behave as an active particle, but together can be much more sensitive to local gradients, and understanding how they react collectively and how that can affect their, how they can rectify motion is something that is qualitatively different to what may happen on the micron or supermicron scale, where the behavior can be more understood at the single particle level. So that was a bit what I thought for the title, but then this requires really understanding first mechanisms, and that's a bit what I will be showing, how there are, to which extent one can exploit or one can try to understand the qualitative different nature of the behavior of interactions and the reaction of some active colloids to the fact that they are out of equilibrium, not necessarily under external field. So depending on how far I go, where do I get, I wanted to focus on one particular case that is standard and that we control well on the lab, which is the use of colloids that are treated on their surface so that they are heterogeneous. They typically catalyze a reaction, and that by electrophoresis or by diffusophoresis allow them to propel. And then the question is, why do they do that? How do we should understand this, and how do they do collectively? And if I have time, then there is a second part where I wanted then to talk about how this is how we directly exploit, understand the activity. The other is, if the environment is complex, is heterogeneous, how these particles interact with the environment and can also react or modify the environment and produce also certain structures, okay? So, as I said, this is a standard. It's a well-known, well-used, I think still very fundamental. There are questions to be understood, but really, so the basics is that these particles, because they are heterogeneous, the catalyzer, say the red part here, decomposes a reactant, and say what I will be showing you, this is motivated by experiments done with decomposition of oxygen peroxide in one case and by decomposing urea. So one can use platinum, for example, or there is a possibility. Now people, for example, also cover this with an enzyme, because then the colloids are more biocompatible. So for some of these biotechnological uses, I mean, you cannot do that with platinum and oxygen peroxide for obvious reasons. But at the end, one has a reactant that is being decomposed in one part of the colloidal surface, and as a result, there will be gradients in reactants and products. If I don't enter, I mean, the details of the chemical reaction itself is non-trivial, but at, say, lowest order, or trying to understand mechanisms, one can think that there is a field phi that is this difference in reaction, in concentration between reactants and products that will not be homogeneous over the particle surface. And if these products are attracted to the colloid, then typically this gradient in concentration over the particle surface leads to a motion by phoresis. And phoresis is a classical subject in colloidal physics or in soft matter, and actually there is a nice review by Anderson that explains all these different mechanisms of phoresis, thermophoresis, diffuso or electrophoresis, how they can be understood on the same framework. And the basic idea, just to set the frame, is that if you think on the surface of the colloid, here being vertical, and the colloid attracts certain species, the concentration of these species perpendicular to the surface will not be uniform, in lowest order, will be an exponential profile that will depend on phi, the potential of attraction. So that's the physical chemistry of the interaction between the chemicals and the colloid. And here chemical potential is uniform, and the pressure also here will be uniform. But the pressure will be the hydrostatic pressure when we move perpendicular to the wall. But if I look up and down, if I put here a gradient, because here say there is more reactant that is being decomposed, this gradient along the wall means that even this prefactor will change along the wall. And as a result, there will be also a change in pressure along the wall because of the local hydrostatic pressure. And if you now have a pressure imbalance by Stokes equation, then that will lead to a velocity profile. So this pressure imbalance generated by the concentration gradient leads to essentially a profile that will be characterized by a characteristic difference in the velocity away and the velocity at the wall, and that's the slip velocity. So that's the basic idea. And that presumes that there is this equilibrium. This is essentially what I write here, and this is typically what we use to understand why this colloids move. However, and I wanted to show this at the single particle level, then I will look what happens with many, but I think this is an interesting example of how subtle the fact that systems are out of equilibrium is, which is, and this is slightly more complicated than the case I've shown you before, and these are experiments done in Samuel Sanchez's lab in Barcelona. Essentially, he considered one of these colloids with this enzyme, and this enzyme, when it decomposes, the products have a charge, typically. So this is in water, so you have ions, which means then that, say, the black part of the colloid that is producing, where the chemical reaction takes place, where the decomposition of the corresponding molecule takes place, will lead to effectively a flux of cations and ions. So one can solve the equations in steady state, but because it's a constant flux, at the end of the day, this is a source. It's a source of ions. And if you have a source of ions that diffuse, actually the profile of the concentration decays as 1 over R. So this assumption that is local and we can forget about the flux that I mentioned in Anderson is conceptually not correct. It's a good approximation in some cases, but it's not correct. Now, in this particular situation, if you think like in these profiles that decay as 1 over R, if the diffusivity of the two ions is not the same, then the prefactor by which they decay, the rate at which they decay is not the same because it depends on the diffusion coefficient, which means then that the charge is not really localized in this Anderson double layer that I was telling you before. Charge spreads over the whole system. So then, even if the process, in essence, that I was telling was diffusophoretic, there is this electrophoresis component that you cannot get rid of, or can sort of like integrate out in the effective potential, as the Anderson picture would tell us. And this then, there is then, what we did was to look at this problem in general. There are, again, it's a fluid mechanical problem. I don't go into the detail. If you have questions, we can sort of go in, but it has no secret. Stokes equation, the diffusion advection equation for the species, and the boundary condition. So here the Cs is co-ions, the anions and cations, and they are produced as a given rate. So there is one dimensionless parameter that is the rate of the production with respect to how quick these ions diffuse. There is another parameter that is this asymmetry in the diffusivity, and actually, I mean, one do the derivation using the Lorentz theorem and so on. One can get to an expression for the velocity that depends on the charge of the colloid and depends on this ratio beta. The ratio, if beta is 1, essentially, I mean, then you will have what here is HOT is the standard Anderson prediction but then there are these two terms that have different parity in beta. And then that means that then again, when you look at values, so this is the velocity as a function of this rate. So if the rate is very low, essentially the red line here is Anderson, so it works. So all these details that the process that I'm describing quantitatively is really not important, but as the rate increases, the rate of production of ions, the activity increases, there can be quite a large, I mean, this is a logarithmic scale, so the difference in magnitude is quite substantial, and not only the magnitude, actually, depending then on the charge of the colloid, there can be cases where, I mean, this is a busy plot, but if you look, for example, here at the red points, then the velocity can change sign, which means that changing the rate of activity, not only the speed, but also the direction. So one can tune the directionality in that way, which is, again, something that in equilibrium or in the limit in which we are in a quasi-equilibrium state is not possible. Actually, if you want visually, what happens when we increase Q is that, I mean, in the low activity region, here the color, I mean, this is half a colloid, and this is the numerical solution of the equations that I was showing you. The color is the amount of salt. So greenish or bluish green is kind of like no charge. So here is really the charge accumulates very close to the surface. This is this double layer limit. That's the Anderson limit. And then as Q increases, what happens is that charge starts being decoupled. Numerically, it's never zero, but it's very, very small. It becomes being relevant outside this double layer. And that changes then there is an electrophoretic component that actually counterbalances the initial motion. That's why it can change sign. So that's for one, simply to say that one cannot take for granted, no? That what we can know in equilibrium, we can just directly extrapolate. We need to understand what's going on to see when this standard description is valid or not. What I wanted to discuss now is what happens when we have a number of these colloids instead of one, but it's another experiment. It's motivated now by other experiments. So these are experiments that are covered where the active colloids are covered with platinum and they decompose oxygen peroxide. And here I will assume that we can use this Anderson picture. There is only one species and a charge is not taken into account. It's purely diffusophoretic. But here I wanted to look at another effect which shows how we can manipulate or make use of this, the fact that these systems are producing these ions, this species out of equilibrium to generate and control how they attract and aggregate in this case and move. And this is an experiment that so basically what they consider in the experiment are these are the active particles, and these are sort of like micron, a bit more than micron size. They have some hematites over there. They tend to sediment. So at the end, what you will be seeing is a monolayer of these particles. So they sediment on a wall, and then we see what happens in the presence of the oxygen peroxide. Now, as it is drawn here, these active colloids that they work with are not perfectly spherical. And it turns out that when they sediment, they tend to sediment with the platinum pointing towards the wall. So basically, a single one of these active colloids will be sort of like decomposing oxygen and essentially sort of like hitting against the wall. Yeah? So you may think that this is a bit stupid because the particle is doing nothing. It's basically hitting the wall, and then because it fluctuates a bit, it will diffuse around. But again, this is producing, it's true, it's doing nothing, but it's decomposing oxygen peroxide. So there is a gradient of oxygen peroxide that, again, goes as 1 over R because it's a sink, if you want, of oxygen peroxide. So now if there is another particle that has a diffusophoretic response to the oxygen peroxide, we'll see a gradient, and then we'll move in that gradient. So then you can have an attraction that is essentially a collective effect, because isolated particles by themselves wouldn't move. You need a few of them. So this goes in the direction of looking into swarming, into how these particles collectively can respond or can behave because of their activity. So what they did is to mix these active particles with silica particles. The silica colloids are passive, but they have, because they also have a silica component. Both species react to the diffusophoretically to gradients in these oxygen peroxide. So if you have 2 of them, and this is experiments with 2 particles, one passive, one active, then what happens is that the relative position decreases. Sorry, yeah, that's the relative position, sorry, the blue one, and the red is the center of mass, I think, and then at some point it will start moving. So when they come together, then they can propel. So they form a dimer, but the dimer is not bound. So if we stop the oxygen peroxide, they will move away. So they are bound dynamically because of this interaction, which is persistent. So again, what we did is we could estimate what is the force that this gradient creates on the silica particle. Silica particle perturbs the concentration, so also affects how an active particle will move. This interaction is not reciprocal. It's not the same. The force that the silica particle distorting the chemical field affects the active particle is not the same than otherwise, but one can compute at the pairwise level the problem of the low Peclet numbers, what is the chemical fields, and integrating over the surface, assuming that the slip velocity is proportional to the gradient, identify these relative velocities, and then we can do now a Brownian dynamic simulations of this mixture of particles. We'll have active-active interaction, active-passive, and passive-passive, okay? So we can solve that, and then in the experiments what they do is, and then we can compare with experiments. So they have this monolayer. This is an image of the monolayer. The active particles are dark, and the silica particles are lighter, and then they actually, something that is useful from the experiment point of view is that the platinum, in principle, catalyzes the oxidation peroxide, but the efficiency with catalysis changes a lot when they are illuminated with the right wavelength, and in the experiments they use that as a switch. So basically, here they have everything ready, and then they illuminate with the right wavelength, and then when they do that, it's quite quickly that they see how these particles approach each other, aggregate, and they aggregate by sort of like trying to, with a certain structure between the active particles and the passive ones. Yes, they can. But not by the wavelength, they do that by the intensity of the light. So the wavelength is what is the switch, is that what is sensitive to, and then depending on the intensity, they can be faster or slower. And then basically what they did in the experiments is to vary the concentration of silica particles and the concentration of these active particles. And basically, if the density is weak, small, then you will have aggregates, clusters that move around. And there are regimes where you have these structures that basically span the system. These are like gels, and at very high concentrations is more like a glass. And as you can see here, if we have more active particles, these gels are like more elongated, while if we move more into the trying to get the equivalent one is a bit more difficult by increasing the silica. That's not the case. This is because these particles, these active particles, because they have some hematite, they also have a magnetic component. So when they come together and join, they tend to form slightly elongated structures. Maybe you could see this here. In the simulations, we also introduce this attraction for comparison, but that's not fundamental for what we see here. So we could identify, in the simulations, so these are the experiments, and then these are the simulations. We could sort of like more or less get the equivalent structures. So at the end, it's possible to get a phase diagram where we have different type of clusters or gels, and we call it gel because we look at the dynamic intermediate structure factor, and we saw sort of like a very low decaying plateau in the angular, in the psi 6 order parameter, because this is a 2D system. So this tells us that indeed we can control and form structures by modifying the activity and relative composition. And I wanted to, how I am doing with time? Because I didn't pay attention. 30. Okay, perfect. So I can finish this and then still explain a bit of the other. I wanted then, so this is just to indicate how activity really, even if, as I said, even if individually they wouldn't do anything, collectively they are able to, or we can manipulate by using I didn't come into this, but it's true that changing the activity, you can also manipulate when these phases or when these different structures appear, and then that's a way to control them collectively without having to go one by one or see exactly what they do. And I wanted to show you also, this is to show that things are sometimes subtle. In the same simulations, experiments, in the same experiments, they also work in cases where the concentration of the active particles was very small. And in those conditions, basically, they could focus on individual clusters. So here you see the cluster, and this is an example of a different power. So with power, they favor that they are larger or not. So they change, so they can observe these clusters at different sizes, and they could measure for example, for the different sizes, the velocity decreases with size, And also, if you look at one of them, the mean square displacement has a t square, so they at some point propel. This may seem, seemed a bit surprising, because in principle, I would have thought that these objects, these rafts are quite symmetric, so there is no need for the particle to move. But actually, when one starts looking into the shape, is not perfectly a circle. And also on top of that, it turns out that when they looked a bit more in detail into the structure, the hematite, the active particles, was slightly off-center. And actually so they could measure, this is exaggerated, it's not that off-center, but it's sufficiently off-center to make it displace. And there was a systematic dependence of this asymmetry with respect to the area. So we understood that this explains why they propel. So it's non-trivial that, in retrospect, you can think that if they start moving, because the particle that is active generates certain motion, it's slightly asymmetrical, the probability that other particles have to get to the cluster, and that we think creates that asymmetry. But something that we found a bit also, yes, sorry.
This cluster stable in the absence of light?
No. Everything is sustained by it has to be active. So light is the way, so let's say, if it can decompose oxygen peroxide by other reasons, then it will be sustained. Light is the practical way to ensure that the rate of production or decomposition of oxygen peroxide is large enough for the interaction to be strong, to keep them together. But if you switch off the light, it's equilibrium, they will dissolve. Yes. So there is no strong attractive interactions between them, between these different colloids.
I thought light is only adding as or used as some catalysis to the decomposition process.
Yeah. So let's say what I understood from the experiments is that in the absence of light, the efficiency of platinum as a catalyzer is so weak that the amount of material that did decompose, or the gradients that are generated, are too weak to really drive a significant attraction between the particles. That's what I understood. Yeah. That's why I said, I don't know if instead of platinum, you could put another or an enzyme that would be more efficient by itself, then you would see that without the light. Yeah. Something that, so as I said, so they had the experiments, and then we could compare with the simulations, the ones that the same model that I described before, and we could compare the power laws, so that was working. But something that we observe is that in the simulations, the aggregates, the rafts that we observed, had a stronger tendency to change their motion. The persistence was smaller than in the experiments, and also when we look at where the particle was, when we start looking at the geometry, the structure of these aggregates, in our simulations, the active particle tended to be in front. So if I look in the direction which the raft is moving, was in the front, while in the experiments was in the rear. So that's something that puzzles us a bit, because we didn't know exactly why. So we look a bit more in detail, and because there is something that I have haven't paid attention to, which is hydrodynamics. As we heard in the previous talk, hydrodynamics is important. These are actually, the system is complicated. You were asking, there is a question of the wall that I was telling before already at the chemical level, from the chemical interaction, but actually, I've been emphasizing that if there is a gradient in concentration, then because of this gradient, there will be this diffuser-phoretic response, but because these particles are on a wall, then it means that if there is a gradient, and actually the material of the wall is the same as the material of the colloids, the wall itself also has a response to these gradients, which then is osmophoresis, it gives rise to an osmotic flow. So in reality, we have both. We have the fact that the particle has this self-diffuser-phoretic due to the gradients that the particle generates, but also these gradients that the particle generates will lead to surface flows. So this raft that is on top of a wall will be subject to a certain flow below and above it. And we initially thought that that would be a second effect in the sense that would simply renormalize whatever is the effective friction, but we decided to look into that, and then again, for that then we have to say that we have to write down, and we did that theoretically again using the reciprocal, the Lorentz reciprocal theorem. We can say, well, we have an object that is force-free, so the force due to this friction plus the force of the force of the phoresis plus the drag have to balance out, and then you can write this down. It's a bit lengthy, but then one can have a prediction about how the velocity will depend on the particle, the object size. In this case, we give an object size and see what happens. And we have actually two mobilities. We have, it's here, yeah. We'll have in reaction to a concentration gradient, there will be a velocity on the solid surface characterized by the diffuse osmotic mobility, and because the particle will have a tendency to move in a gradient concentration with a diffusophoretic mobility, we have these two coefficients. And actually, because both flows respond to the same gradient, actually, this chi here is like the gradient with other terms. Actually, the velocity of this object depends on this gradient and depends actually on the difference between these two mobilities. So in a way, the phoretic flow and the osmotic flow have opposite directions. So this is simply telling us that because there is this competition, we can expect that since the solid wall and the colloid are made of the same material, both mobilities will have the same sign. So it's a question of which one is stronger. So the fact that it goes opposite to the direction that would assume the phoretic mobility suggests that the osmotic mobility of the substrate is higher, and that then determines that it's moving in the opposite direction. So that's told us that if we want to understand properly, not the kinetics, I think it's captured already with this phoresis, but if we want to capture properly how these rafts move and interact among themselves, then one cannot leave aside also the fact that they move and generate flows and interact also through these flows. So that's something to look in the future. And in the remaining sort of like 5 or 7 minutes, yeah? Now I wanted to change gears and then look at another aspect in how these active particles can interact collectively with a medium that is a bit more complicated than a solvent. A Newtonian solvent is always complicated, but this is a medium with a structure. And I will do that by, again, looking at simulations. In this case, there are no experiments so far. And I will also, for the time being, disregard hydrodynamics. So what we looked at, in principle, is something that is relatively straightforward. So we consider a binary mixture, a fluid binary mixture that can phase separate, in which we have active colloids. And we did that by assuming or considering that now the colloids behave as active Brownian particles. So they will move with a characteristic velocity that will depend on their orientation. And then there is a colloid-colloid or particle-particle interaction. They cannot overlap. And then they will also interact with the binary fluid. Actually, we do that by the binary fluid is simulated through coarse grain by a field, psi, that will be +1 in one phase, -1 in the other phase. And we solve, essentially, it's like a Ginzburg-Landau free energy, and we solve the Canhillier model for it. So this is a continuum field. Particles are individual particles, but each particle has a size. The field can go inside, and then we introduce a coupling free energy between the particle and this field. That's how it will prefer to be in one phase, or in one liquid or the other. And then associated to this coupling free energy, there is a force. And in principle also, this direction of motion will diffuse around in 2 dimensions. It's characterized by an angle, so it will have a rotational diffusion. And also then there may be a coupling, in principle, between different colloids, between different particles. There could be an active torque that we will not introduce, and can also be a torque with the order parameter if it's not, if there are gradients there, okay? So all this comes from a free energy, so in a way it has a well-defined thermodynamic limit, And what we do is we solve this hybrid. So we solve in a lattice this order parameter, which is a field, and then in this field that is moving, at the same time then is coupled to these particles that are moving around. The motion, the level of activity of these colloids, well, because I mean, as I said, they self-propel at a given characteristic self-propelling velocity, and the inverse of the rotational diffusion is like time in which they sort of like lose the memory in which they are moving. We will define as usual, and it's usually done, this active Peclet number, which is a ratio between these two times. So so Peclet large means that the particle can displace long distance compared to its own size before it will rotate away. So what happens if I now take this binary mixture at high temperatures? Now I do a quench, and then so it starts from an initially homogeneous state, and then it will phase separate. And then if I look at the characteristic domain size, what I see, what I will be showing you is for a 20% area fraction of these suspended particles. Then typically it coarsens with a power a third if the particles are not very active. And at long times, well, you will have complete phase separation. I will be showing you results when the particles, the colloids, are soluble in the white phase. Okay? Again, that's controlled through that free energy I was mentioning before. Now, what we observe is that as we increase Peclet number, there is then a regime where the coarsening is arrested, and what we see is basically a state where the active particles have broken up the other phase into, this is for one particular case, into domains that are relatively spherical. So this is more like an emulsion, if you want. So active particles are able to emulsify a binary mixture. So this is 5 minutes? Yeah, okay, I'll try to finish soon. So in order to look a bit more in detail what's going on, rather than looking at this, we started with an initial condition where the two phases are completely phase separated. So now, if we have complete phase separation, the domain size, if a Peclet is small, keeps being large, so nothing happens. And indeed, as we increase activity, then the typical domain size decreases. We more or less end up in the same place. So what we see as the final state is, I think it's a reasonable, really well-defined steady state. And indeed, there are, so this, at very low activity, the active particles stay in their phase. At intermediate Peclet numbers, then there are these domains. At even larger Peclet numbers, then there is another state where domains stay there, but they are smaller. And then eventually, when particles have enough activity, at the end, they can cross all interfaces, but then they end up accumulating in the wrong phase. That's the phase where they are not soluble. And this is essentially because in the phase that they are not soluble, because they have a stronger repulsion energy of forces, their actual velocity decreases, and active particles tend to accumulate in regions where their velocity is smaller. So they are trapped there dynamically in a way. So we computed the typical domain size, and as I said, in this region 1, this is the system size. Then we have this decay. Actually, the decay of the domains goes as 1 over the Peclet, and this can be understood if you think that what happens is that at the end, the binary mixture wants to phase separate, so there is, if you want, an energy associated to it that is because of the surface tension. And then, on the other hand, the active particles have an active pressure. So if we sort of like assume that this is the balance between these two pressures, the capillary pressure against the active pressure, actually the prediction is that the radius of the domains goes as 1 over Peclet number. So at least the scaling can be understood as a way of balancing these two. But maybe it's more clear what I say if I show you a few movies. So this is low Peclet, and yeah, you see that basically the domains stay being separated. When we go to these intermediate cases, then now activity is able to break up the gray phase into a state, an emulsion, where domains have a quite relatively well-defined size. You could even wonder whether they want to have a certain structure, order. And then at some point, so here essentially the active particles still stay mostly in the soluble phase, it's difficult for them to cross because there is the cost of the energy, the capillary energy. Now, there is this intermediate, what I will show you, where they can go through start being able to go through the gray phase. And here, it's not just that the domains are smaller, it's that they are much more dynamic. You will see that now, so we classify this as two types of active emulsions. Both are active and different from passive ones, but here is really more, here we don't have a balance that will tell us what's going on, but we see that particles can go in and out these gray domains. And now here, when they have a large enough activity, then essentially they go to the gray phase. They can go there because then they can surmount easily the capillary forces and then stay in that phase because they move slowly, more slowly there effectively. Okay, so I think that's an example of also how collectively, because that, again, you cannot do that at individual level, but this active pressure can create new types of structures, in this case, when you have a medium that is more complex than just a homogeneous medium. And that's, I don't know, I mean, it's something that is still, I don't know to which extent that's relevant or not, but there is this now a lot of interest in biophysics in understanding how inside cells one can sustain structures that are liquid where surface tensions are small, when these liquid-liquid phase separations that are arrested. And actually this shows that activity may play an important role in determining how these intermediate structures that are metastable, they are not equilibrium, can be sustained dynamically. And actually this shows that there are different regimes by which, or different mechanisms by which such type of emulsions can be sustained. So I think I'll finish here. There was now a last part showing that also activity can create spontaneous chirality, but I will try to skip that, sorry, because I have to go through all these movies, yes. Simply to say that I hope show you the importance of medium and the chemical interactions and how that can lead to new scenarios to manipulate these clusters dynamically, both in the case of these self-diffusive colloids, or also in the case of the inhomogeneous media, how we can have or promote, stop these phase separations and generate new non-equilibrium structures. This has been done in Barcelona. The theory, I mean, most of the simulations by Javier Diaz, a postdoc, and Sergi Granados, a student, and as I mentioned, the experiments were done in the group of Pietro Tierno with Dola Chai and Elena. And thank you very much for your attention.
Thanks, Ignacio. Questions? Kabir.
So very interesting. So in the ABPs, when you have this growth of this length scale for different Peclet number, you seem to have a T to the 1/3 law, which corresponds to the Lifshitz sort of growth. But that's equilibrium, if I'm not mistaken.
Well, I mean, that's, so I couldn't call it, this is coarsening.
Coarsening, yes.
Controlled by diffusion against interfacial energy in that sense. So yes, so it's equilibrium in that sense. And indeed, if you think, we see this coarsening when Peclet is not very large, or even when it's large initially, we see that probably because domains are small and can start growing, and then at some point it's arrested. So I would say the activity part of this pressure generated by the particles acts precisely at arresting what this underlying kinetic phenomenon.
And when does the crossover happen? That's governed by the active time scale.
Yes. So what I didn't put here the expression. So we have this estimate that we say that the arrest is controlled by a balance between the active pressure, and the active pressure is proportional essentially to the velocity and to the number of particles against the Laplace pressure. That sets a scale. And then that will tell you when you will be arrested. So the arrest is not a question of time, it's a question of length scale when these two pressures are balance each other. If the domains are very small, capillary pressure is large enough to favor growth, and then at some point the activity is able to arrest it.
So the critical droplet size, because that will, the Laplace pressure for the that size droplet will match.
Yeah.
I see.
Yeah.
Okay, thanks a lot. Yeah.
I have a quick question. So if suppose the active particles had a tendency to flock, and now you put them into this complex environment. So have people thought about this?
I mean, no, not that I'm aware of. Actually, in the part that I didn't talk about vorticity, there we look at it because that's very important to promote chirality in confinement. But that's a natural follow-up to see now what. That's why I said I show you the simplest case, no? But that's the fact that this happens now. It definitely makes it relevant to try to see now how other aspects of activity influence, interact with this mixture. Yeah.
Thanks for a very interesting talk. So you showed us a phase diagram where you had 3 different kinds of gels, gel 1, 2, 3. So how do you differentiate between the 3 phases and which quantity is responsible to create a differentiation between the...
To go there, maybe so in short, these are related to the fact that the active particles are magnetic. So at the end, so what we call gel 1 and gel 2, I mean between 2 and 3, honestly, so how we distinguish? We have a measure about the asymmetry of these domains.
Okay.
So basically, let me see if I go to the next one. Yes, here we can see where gel 1, so gel 1 is here. That's what I say. These are gels where there is a majority of the silica component, while the others are gels where there is a majority of the active component. The active component, because of the magnetic interaction, has this elongated, they have a tendency to form these very elongated sort of like arms. So if you try to now measure a bit the dispersion along a characteristic length, one is more asymmetric than the other. Okay. It's a morphological measure to that. Between 2 and 3, honestly, I think the 3 is when we are moving in the active component, when we lose that, when the asymmetry becomes weaker, so it's a bit more isotropic. But I would need to look back again. I was always a bit doubtful about the 3. So 1 and 2 for me is clear because of this different way to aggregate the particles, especially the active ones.
Thank you.
Basa first, then Abhijit.
In the last part of your talk, the initial configuration, if I understand correctly, you had a white phase and a gray phase, and white had active particles and the gray had passive?
No particles.
No particle.
No particle. Okay. So the active particles soluble. If the active particles are not active, then they are, anyway, thermodynamically, the particles are soluble in the white phase.
Okay.
And then, but all the particles are the same. All are active. And then when I changes the degree of activity of these particles.
Sure, yeah, thanks.
The active pressure is sort of dependent on the velocity, the activity.
Yeah.
What's the role of the length scale of the active particle or its mass?
So there is no mass. So this
in this
is
case,
Ovedam.
okay. There's no
in
inertia.
this case there is no mass.
Okay.
So there is a length
scale
which is the persistence length of the
Okay.
particles, but that's again encoded on this active velocity. They go together. They are not independent.
More questions?
That's another interesting direction, which is also an ongoing issue. That's a bit more formal, but the role of inertia on active particles, which then would be specifically the mass, is always something that tends to be left aside, and exactly when or how it applies, I mean, there are some works, but I don't think it's completely settled, so that could also be interesting.
Yeah.
Nice talk, Professor. So that last part you just flashed, that I was wondering that spontaneous chiral symmetry breaking. So will you please mind to just briefly say that phenomenon, what you get there?
Well, just very briefly. So basically, what we, if I have an image, I can tell you. So essentially, the idea is we have the same type of particles now on a phase which will form domains, spherical domains. We do that, I mean, this is for a block of polymer, but the point is that it's a soft spherical circular confinement. Now, if the active particles, if they are, in this case, they were soluble in the gray phase, activity by themselves will favor that they tend to go to this interface and stay there. What we saw is that, in this case, that's where we were introducing the alignment. If we introduce an alignment between the particles, when they come there, they have this tendency to move, to be sort of like perpendicular, but if by a fluctuation they sort of like start rotating or being aligned, moving, say, parallel to the wall, to the interface, then they have a stronger tendency to stay there for longer. So they still can come in and out, but statistically they tend to accumulate into what we call trains in an intermediate region of persistence, and that then gives a spontaneous breaking because there is nothing that is chiral in the interactions themselves. Each domain can sort of like obviously rotate in different direction, and that's, say, how they create this vorticity.